Illustrating a developmental network is very helpful when determining the pathway of gene regulation among a species like the lizard, the thorny dragon. In this video, we will be discussing a hypothetical developmental network based on the trait of external spikes among thorny dragons. Regarding what genes are regulated, we need to determine whether or not the DNA-binding proteins have access to the lizard's DNA. The chromatin, which is a complex of both DNA and protein in eukaryotes, can be altered. Alterations of this chromatin structure can cause protein binding to become more difficult and can result in a change of gene expression. An example of this is DNA methylation. DNA methylation is when additional carbon methyl groups add to the DNA, and in turn, this blocks certain genes from being expressed, leading to less DNA transcription. DNA methylation is considered an epigenetic change since the gene change does not occur at the sequence level like a mutation would. Instead, this gene change is due to the random addition of a molecule onto the DNA. Based on the wild type presented on the thorny dragon, we can determine what consequences in gene expression would arise if we were to have various mutants all lacking different aspects of the developmental network. So, blue activates the expression of orange, which blocks the expression of green. Green, when activated, will block red, which can activate yellow, resulting in spike expression, seen here. Therefore, we know that normally under wild-type conditions, spikes will be observed. Once orange blocks green, here, nothing is available to block red. This will result in the activation of yellow, which activates spikes. In a mutant lacking orange, green would never be repressed. If green is active, it will repress red, which will not be able to activate yellow. Therefore, this mutant will not have spikes expressed. In a mutant lacking green, red will not be repressed. The activated red can now activate yellow, which will then activate spikes. This mutant will express spikes. In a mutant lacking both green and yellow, spikes will not be expressed. If green is gone, red can activate yellow, but without yellow available to become activated, the gene will never be expressed. In a mutant lacking blue and red, the lizard will not have spikes. Although blue isn't there to initiate the blockage of red via orange and green, without red, the yellow will not be able to activate spikes at the end of the network. By using a developmental network, we can see how certain genes are activated in cells based on where they are needed. So, for example, due to its environment, the lizard may need spikes to protect from predators, therefore the genes would all transcribe. But because all body cells are a part of the same genome, this developmental network could also be shared with the genes that work on internal skin. Blockages like these would occur, ensuring spikes did not form. Again, regulation through DNA methylation could be a cause of the selective response and activation in thorny dragons.